Hello students, welcome to EPG Pathshala. I am Dr. Rajesh Gautam from Dr. Harishingar Vishwavidyalaya Sagar, Madhya Pradesh, and today I am going to discuss about the module entitled Genetic Structure of Human Populations under the paper Population Genetics. So dear students, let us first we know what we are going to learn in this module. To understand, first we will understand the meaning of genetic structure of population, then how to describe the genetic structure of any population, then we will also understand the approach of genetics, then consequences of population division. You will also understand hierarchical population structure and reduction in heterozygosity. I will also tell you technical comments on genetics, genetic structure of human population, then you will know effect of population structure and agent and sources of genetic structure change, importance of population structure and dynamics. After reading this module, you shall know how to quantify the genetic structure, and these are the two parameters by which you will know to quantify the genetic structure. One is fixation index and genetic distance, and second is model based clustering algorithms. So let us start. Genetics is a term derived from Greek word gen, which means to become or to grow into something. This term was introduced by Watson in 1906. The genetics is that branch of biological science which deals with the transmission of hereditary factor from one generation to next, and also the ways in which express themselves during the development and life of an individual. The study of molecular genetics deals with the biochemical nature of heredity, specifically DNA and RNA. Genetics focus on the biochemical nature of heredity, including the structure and function of genes and other DNA sequences. Genetic structure is a pattern in the genetic makeup of individual within a population. A very good definition of population structure is when population have deviations from Hardy-Weinberg proportions or deviation from panmixia, if there is inbreeding or selection, or if migration is important, then population can be said to be structured in some way. There are two ways of describing genetic structure. In this context, let me first understand genotype of frequencies. Genetic variation in population can be analyzed and quantified by the frequency of alleles. Genotype frequency in a population is the number of individuals with a given genotype divided by the total number of individuals in that population. In population genetics, the genotype frequency is the frequency or proportion, that is f is less than 0, f is greater than 0 and less than 1 of genotypes in a population. Although allele and genotype frequencies are related, it is important to clearly distinguish them. Genotype frequency may also be used in the future to predict someone's having a disease or even a birth defect. It can also be used to determine ethnic diversity. Phenotype of frequencies. Phenotypes are based upon the content of the underlying genes comprising the genotype, the expression of those genes in observable trait is also to varying degrees influenced by environmental factors. Approach used to investigate phenomena in population genetics and many other biological disciplines can be generally separated into three basics. The traditional empirical approach in population genetics comprises extensive observation of the genetic variation of a particular gene or genes in a population or populations, perhaps over time, and the measurement of related factors such as environmental patterns that may influence this genetic variation. These data may provide associations between the patterns or levels of genetic variation and other factors, thereby suggesting potential problem for further study. The genetic variants used initially in these empirical investigations included morphological variants, blood group, polymorphism, and chromosomal inversions, and then starting in the 1960s, allogeneic variation, that is genetic variation in enzyme and protein. Some of the classic example of genetic polymorphism such color that need to be clarified for a comprehensive understanding of the factors influencing their frequencies. In recent years, similar empirical examination have focused on DNA sequence variation mainly between different species but also between individuals within the same species. Generally, only experimental tests can provide support for hypotheses developed from empirical data about the effect of particular factors on levels and patterns of genetic variation. Traditional experiments are exemplified by the moving or translating of a population to a new environment and comparing them with non-transplanted population to examine the significance of an environment on genetic variants. However, in recent years, the definition of an experiment in evolutionary genetics has become broad and includes, for example, comparison of DNA sequences between organisms or genes that have different histories, function, or other characteristics. The consequences of population subdivision and other types of random mating. To understand this aspect of population structure, we have hierarchical population structure. Let me understand what is hierarchical population structure. A population is said to have a hierarchical population structure if the subpopulations can be grouped into progressively inclusive levels in which at each grouping the next lower level are included within the next higher ones. To consider a concrete example, imagine we interested in the population structure of a widespread species of freshwater fish. The lowest population level consists of a local interbreeding population of animals within a stream. A stream may contain more than one such local population. The next higher level in the hierarchy may be the organization of stream into groups feeding the same river. Another higher level may be watershed within continent. The aggregation of subpopulation into progressively more inclusive groups may continue for as many levels as inconvenient and informative. It is inevitably somewhat arbitrary how the groups at each level are combined to form the next higher level in the hierarchy. The objective of the classification is informative. One tries to group the subpopulation in such a way as to highlight the genetic similar and differences among them. If so much of migration of fish among subpopulation that all members of the species constituted essentially a single random mating population, then there would be no need to define a hierarchical population structure because it would be uninformative. Most of the organisms do have significant population substructure. What is reduction in heterozygosity? Let me understand it. One of the important consequences of population substructure is reduction in average proportion of heterozygous genotype relative to that expected under the random mating. Within each subpopulation, there is random mating, and the genotype frequencies, though extreme, still satisfy the Hardy-Weinberg principle. Therefore, within any one of the subpopulation, in the frequency of heterozygotes equals the expected with Hardy-Weinberg equilibrium. Now, let me understand some technical comment on genetic structure of human population. Estimates of genetic variance components depends on the type of marker used, the definitions of geographic regions, the populations sampled within these regions, the relative sample size from the populations, and the way in which information is combined across loci. For microsatellite marker, estimates also depend on whether the quantity whose variance is partitioned is an allele size variable for an indicator variable for allelic presence or absence. A main purpose of our variance component estimation was to provide insight into the fine scale population structure analysis, because the structure algorithm uses only to identity and non-identity of alleles. Descriptive statistics that employ allelic indicator variables are more appropriate for understanding the dependence of structure based inferences on the level of difference among groups than are statistics that use allele size. Scoffier and Hamilton performed a complementary variance component analysis demonstrating that when a subset of our data corresponding to is studied using allele size as was done in, similar estimates were obtained. They are smaller within population variance component compared with that is consistent with the smaller estimate of in relation to microsatellite studies that used indicator variables. Previous indicator based studies of microsatellites and other markers have not all been in full agreement. A difference in the nature of the variable cannot be the sole source of differing estimates. First, the homogenizing effect of the higher mutation rates of microsatellites in contrast with those of other markers probably explains some of the difference of our results from non-microsatellite indicator based studies. Second, consistent with past observation, the high fraction of tetranucleotide loci in our data contributes to higher within population variance component estimates than are seen in dinucleotide studies. Third, the estimates vary considerably across sampling scheme within regions and in several cases, past microsatellite samples that included multiple groups per region used populations that are among the most differentiated of the groups. Any estimate computed with the Bell separated populations that contribute to the 83.4% within population variance component obtained by Escoffier and Hamilton should be regarded as a lower bound groups in data. Any estimate computed with the Bell separated population that contribute to the 83.4% within population variance component obtained by Escoffier and Hamilton should be regarded as lower boundary. So, dear students, after knowing technical comments on population structure or genetic structure of human population, let me come to know effect of population substructure. The multiplication in equation makes a number of assumptions about human populations. First, that a Hardy-Weinberg principle hold for each locus. Second, that each locus is statistically independent of others so that the multiplication across loci is justified. And third, that the only labels of population substructure that is important for DNA typing is that of race. Critics of multiplication rule argued that genetically important subpopulations need not coincide with racial designations. For example, the term Hispanic includes a mixture of different subpopulations with variable amounts of Spanish, Native Americans, American Indian and African ancestry. Similarly, there are potentially important different allele frequency among Caucasian population and black population. Defenders of the multiplication rule argued that population substructure would have a relatively minor effect on the final outcome of the calculation and that what matters most in is not a high degree of accuracy, but rather a general sense of whether a particular multilocus genotype is rare or common. Now, to explain the change in genetic structure, we have a flow diagram through which we can understand source of variation in genetic structure and agents of change. So, the source of variation of genetic structure are mutation and recombination, and under recombination there is crossing over and independent assortment, whereas the agent of variations include natural selection, genetic drift, migration, mutation and non-random mating. Now let me discuss about agent of change of genetic structure. First, let me know mutation. Mutation is the ultimate source of genetic variation in the form of new alleles. Mutation can result in several different types of change in DNA sequences. These can have no effect, alter the product of a gene or prevent the gene from functioning. Through this diagram, you can understand how that genetic mutation operates in changing genetic structure. The second agent of change in genetic structure is natural selection. It is the fact that some traits make it more likely for an organism to survive and reproduce. Population genetics describe natural selection by fitness as a propensity or probability of survival and reproduction in a particular environment. The third agent of change in genetic structure is genetic drift. Genetic drift is a change in allele frequencies caused by random sampling, that is, the alleles in the offspring which are random sample of those in the parents. The fourth agent of change in genetic structure is migration. Many populations are not completely isolated and exchange genes with each other. Individual migrating into a new population may introduce new alleles into the gene pool and alter the frequencies of existing alleles. This can further be understood by this diagram. Now let me know how non-random mating operates and change genetic structure. Many populations do not mate randomly for some trait, and when non-random mating occurs, the genotypes will not exist in Hardy-Weinberg equilibrium. Positive assortative mating, individuals with similar phenotypes mate preferentially. Negative assortative mating, in which phenotypically dissimilar individuals mate more often than randomly chosen individuals. Neither affect the allele frequencies, but both may affect the genotypic frequencies if the genotypes are genetically determined. So dear students, now let's we come to know importance of population structure and dynamics. It tried to conceptualize what a current or indeed ongoing global head count of whites would look like. It is imperative to keep in mind the age structure and reproductive profile of whatever population exists, as well as the dynamics of rapid change along key vectors. Nothing is stationary or mirror the past. Habitual patterns of though quickly become outmoded without anyone being aware of it. Contemporary demographic statistics conceal racial information. Elites are obsessed by race, and particularly with accelerating in any way possible the decline and disappearance of the white race. But meaningful data on ethnicity do not exist. It is the only major variable not regularly measured or recorded by demographers. If such figures were available, they might be jeopardize indefensible policies given replacement migration, exceptionally high non-white birth rates, sub-replacement fertility among whites, and widespread culturally encouraged hybridization with non-whites in all formerly white homelands around the world. The opposite end of the national age spectrum everywhere now feature a predominantly white elderly population and an increasingly non-white young population. As a consequence, former first world nations are composed more or more of non-whites and hybrids. This process is taking place with lightning speed. For better understand or to better understand the crisis, it is helpful to employ a demographic tool known as population pyramid. A population pyramid is a graphical representation of the distribution of age groups, usually by country or region, shaped like a pyramid when populations are young and growing. There are two basic shapes of population pyramid. In classic shape of population pyramid, it is visible in this diagram that it is a population pyramid of a young, rapidly growing population with a high birth rate. Here, the base of pyramid is broad and top of pyramid is narrow like a natural pyramid. You can visualize here an inverted pyramid in which the base is narrow and top of pyramid is broader. It is a pyramid of a stable replacement level population characterized by low infant mortality, little or no demographic growth expectancy. Now let me know how to quantify population structure. There are two ways of quantifying population structure. One is by fixation index and genetic distance, that is FST and genetic distance. Non-random mating in a population with substructure has two consequences. First, preferential mating between individuals from the same subpopulation is a form of inbreeding and has the effect of reducing genetic diversity in the overall population. Second, as the subpopulation experience independent genetic drift, allele frequency at genetic marker tend to diverge. FST is defined as the decrease in heterozygosity among subpopulation relative to the heterozygosity in the total population HT. So, the FST can be written like this: HT minus HS over HT, where HS is expected heterozygosity computed under the assumptions that mating is random within each subpopulation, that is Hardy-Van Wert equilibrium, while HT is analogously computed assuming random mating in the entire population without population structure. Secondly, the population genetic structure can also be quantified by model-based clustering algorithm. Cluster analysis refers to a large family of approaches whose goal is to simultaneously define subsets called clusters and to assign observational units into these clusters, so that members in the same cluster are similar by some criteria. For comprehensive survey of clustering approaches in the context of inferring genetic structure, the data usually consist of individuals genotyped at multiple genetic markers, example given restriction fragment length polymorphism, RFLPS, microsatellites or SNPs, which are also known as single nucleotide polymorphism. In the discrete population model, all alleles in an individual are assumed to be drawn randomly from one of the subpopulations according to a set of allele frequencies that are specific to each subpopulation. So, students, now let us summarize that what we have discussed in this module. Here we conceptualize the genetic structure of human population. We understand the genetic structure by genotype and phenotype frequencies. Here I also illustrated the approach of population genetics. Then I told you consequences of population subdivision and hierarchical population structure and reduction in heterozygosity. Then I explained you the technical comments on genetic structure of human population, effect of population structure, source and agent of change of genetic structure. These are mutation, migration, natural selection, genetic drift and non-random mating, importance of population structure and dynamics, and finally you also come to know how to quantify population structure by fixation index. Thank you.